Bacteriophages, Our New Greatest Ally by Sarah Ash and Preeti Nagasuru. Antibiotic resistance is becoming an ever-increasing problem in this day and age. Today, we are presenting bacteriophages as an opportunity for a new antibiotic alternative. Unlike what their name suggests, bacteriophages are not bacteria. They're actually viruses. Unlike other viruses, though, bacteriophages only target bacteria. Although the number of different bacteriophages is estimated to be at around 10 to the 31st, each different phage targets one species of bacteria or a few smaller strains. This means that different bacteriophages can be used to treat different bacterial infections, meaning that bacteriophages can treat a wide range of infections. Like other viruses, bacteriophages start off their reproduction cycle by attaching to a host. There are two different types of cycles that bacteriophages use, the lytic cycle and the lysogenic cycle. The lytic cycle begins with the attachment of the bacteriophage to the bacteria. Next, the bacteriophage injects its DNA into the bacteria. Once the DNA is inside, replication takes place as well as protein synthesis. This allows the different parts of the bacteriophage to be produced inside of the cytoplasm. These separate parts then assemble to make new bacteriophages. Later in the cycle, the bacteriophages express a protein that allows the stability of the cell wall to become unstable. Like the lytic cycle, the lysogenic cycle begins with the bacteriophage's attachment to the bacteria. But then, unlike the lytic cycle, which immediately begins to create other bacteriophages, the lysogenic cycle allows integration, where the phage DNA recombines with the bacteria's actual chromosome. Depending on the conditions, the bacterial cell with the prophage will either A, replicate and create cell division, allowing the amount of bacterial cells with the prophage to multiply, or B, induce the lytic cycle, where the prophage exits the chromosome and starts to begin the lytic cycle, meaning that it creates the proteins that the bacteriophage will need to create more bacteriophages to cause the cell lysis. Bacteriophages have been used as a treatment for humans for a very long time. The estimated beginning of bacteriophage treatments began in 1919. Prior to this, though, there were multiple cases where scientists thought they had found something that had antibacterial properties. For example, in 1896, Ernest Hankin suspected that there was antibacterial activity in the waters of Ganges in Jumna. He suggested that an unidentified substance was responsible for the limitation of the spread of cholera. In fact, a scientist called Frederick Twort suggested that maybe the phenomenon was due to a virus. Unfortunately, though, due to lack of funding for research, he cannot further pursue. Some of the first real experiments done with bacteriophages were completed by a scientist named DRAWBACKS. Using samples taken from French soldiers with dysentery, he created petri plates which showed small clear areas, which he eventually named plaques. Because of this discovery, DRAWBACKS titled himself to be the discoverer of bacteriophages. Soon after this discovery was his first clinical trial use of bacteriophages on a 12-year-old boy with severe dysentery. The boy recovered within 2 days, and shortly after that, even more patients received the treatment. All of them recovered very quickly. Shortly after that, in 1921, a study by two scientists showed that bacteriophages were significant in treating staphylococcal skin disease. Since then, many studies have shown that bacteriophages are useful in treating many different kinds of infections, including bacterial dysentery, infections of the skin and nasal mucus, suppurative skin infections, lung and pleural infections, and other various infections. In fact, many people in the United States who are on the brink of death due to bacterial infections have been saved by bacteriophage treatment. There are definitely pros and cons of using bacteriophage treatments, though. For example, as I said earlier, there are estimated to be around 10 to the 31st different types of bacteriophages, each bacteriophage only targeting one species of bacteria or a few strains. This means that you need to know exactly which bacteriophage you need to treat an infection, even after you determine what bacteria is causing the infection. This means that so many cases will be highly specialized and individualized, and it would be hard to cultivate and create a bacteriophage treatment for everyone because of the amount of work it will take. It also complicates the incentive for pharmaceutical investment. Additionally, bacteriophages are not immune to resistance. Luckily for us, though, it has been observed that if a bacteria gains resistance to bacteriophages, it loses resistance to antibiotics, meaning that even if bacteriophages does not completely solve the problem, it is possible that the antibiotics could come into play and help. Now, as for concerns to safety, there need not be. Bacteriophages only attack bacteria's cells, hence the name bacteriophage. Prefix bacterial, meaning bacteria, phage, meaning to eat or consume, meaning bacteria eater or bacteria consumer. Additionally, bacteriophages are highly specialized, and due to the difference between bacterial cells and human cells, it will not attack human cells. The pros of bacteriophages. Basically, antibiotics attack all bacteria, meaning even the good bacteria in the gut are killed. Bacteriophages only kill the target bacteria, meaning that the good bacteria in the gut can flourish. This is key because good bacteria help people stay healthy, and by killing the good bacteria, the bad bacteria have more space and resources to multiply. This could give way to recurring antibiotic-resistant bacterial infections. Also, unlike antibiotics, bacteriophages can mutate and evolve just like bacteria. This allows sort of a more natural chase, unlike antibiotics, which have to be manually changed and bettered to keep up with bacteria. Bacteriophages, unlike antibiotics, have been naturally killing bacteria since they've both existed, and they've been very successful with what they do. Additionally, antibiotics are becoming less and less effective over time, and it's becoming more and more complicated and time-consuming and fun-consuming to discover more antibiotics. Bacteriophages are definitely the way to go because they're already here. We just need to learn more about them so we can utilize them.